Okay, so last time we finished with the stomach. So let's now look at the small intestine and look at all the events which are happening here. So in the small intestine, you do have digestion. You have digestion of all the main food groups, but the main function of this is the absorption of nutrients. Okay, so all your food is broken down into the smallest absorbable particles, and that's what is done here. By the time food actually reaches the ileum, remember the small intestine was made of duodenum, jejunum, and ileum. By the time it actually reaches the ileum, most of the absorption has already been done. Okay, so and you know that in the small intestine you have the villi and other things which increase the surface area. So you can see why it increases the surface area, because of this function of absorption. And as I said, all the main food groups, not only fats, proteins, and carbohydrates, but you have nucleic acids and other things. Those also get digested and absorbed. And let's see how the events begin. So from the stomach, remember the pH was acidic and the food was broken down into a pasty kind of fluid which was called chyme. And that chyme is acidic. It enters the duodenum, and remember 3 ml at a time. So once it enters the duodenum, it sets about a chain of events. The first is it stimulates the duodenal mucous membrane. So it causes secretion from the mucous membrane there, or the epithelium. And here the first idea is to neutralize the acid which is produced, because you don't want the duodenum to get digested. You don't want any injury to the duodenum. So the epithelium has, if you remember in the small intestine, you have lots of goblet cells. And also in the duodenum, you have these submucosal glands called Brunner's glands. So both the epithelium and the Brunner's gland, they produce a mucus-rich secretion with plenty of water. So that helps to neutralize the acid which is coming from the stomach. And second, if you remember, the epithelial cells, they had microvilli. So in the villus, and if this was an epithelial cell, and I take just one of the cells, you remember it had this microvilli, and they looked like a fuzzy brush. And this was known as the brush border area. So this produces enzymes. So apart from just water and mucus, the brush border of these cells, of the epithelial cells, they produce what are collectively called brush border enzymes. So they also add to the intestinal juice secretion. These brush border enzymes, they digest carbohydrates and proteins. And, they have names like maltase to digest maltose, lactase to digest lactose, things like that. So their names are such. So they digest carbohydrates and proteins. So that's the first thing that happens, intestinal juice secretion. Second is it causes the duodenal mucous membrane, the epithelial cells. Remember, everywhere you have these enteroendocrine cells. That means endocrine cells which are present in the digestive tract. Those secrete 2 hormones called cholecystokinin and secretin. So these are released by the intestinal epithelium. And what is the function of these? Secretin goes to the liver and causes bile production and transport of bile through the various ducts. Cholecystokinin remember in the beginning, bile goes into the gallbladder to get stored and concentrated. So now from the gallbladder you need bile to come down. So cholecystokinin causes the gallbladder to contract so that it can squeeze out the bile. And then the hepatopancreatic sphincter, or what was known as the sphincter of Oddi, which was guarding the opening of the bile and pancreatic duct, you want that to relax so that bile can enter the duodenum. Okay? So this is the function of those 2 hormones. Then at the same time, these hormones also cause pancreatic juice secretion. So not only do they cause bile production and gallbladder contraction, but they cause pancreatic juice secretion. So cholecystokinin and secretin both go into the bloodstream. From the bloodstream, they travel to the pancreas and they cause pancreatic juice secretion. And the secretion produced by them is a little different. Cholecystokinin causes the secretion to be extremely enzyme rich. So it has a lot of proteases, lipases, amylases, whereas secretin causes this acinar cells to produce more bicarbonate. And the function of this bicarbonate again is to help to neutralize the acid. So not only is the acid neutralized by the mucus which is produced here, but quite a lot of the acid is neutralized by the bicarbonate. And then vagus, remember, so these were hormones which brought about activity in the small intestine. The vagus, parasympathetic part, also causes weak contraction of the gallbladder. So this is the main thing that is happening for production of the various juices and the enzymes. So here let's take a look at this. So here you can see the first is that chyme enters and causes secretion of intestinal juice, which is not mentioned in this chart here. And that intestinal juice contains mucus, water and brush border enzymes. And then chyme, which enters the duodenum, it's acidic. It causes the enteroendocrine cells to release cholecystokinin and secretin. They enter the bloodstream, you can see here. From the bloodstream, they go to the pancreas, where secretin causes bicarbonate-rich pancreatic juice and cholecystokinin causes enzyme-rich pancreatic juice. And then secretin also travels to the liver, and the liver produces bile salts. Remember, in bile there was bile salts which are produced. That when that enters the duodenum, those bile salts actually cause a positive feedback mechanism. So both bile salts and secretin, they cause the liver to produce more bile, and they cause the bile to be transported into the duodenum. Cholecystokinin causes this gallbladder to contract, because here in the gallbladder, bile, which is not normally traveling into this part, when it's produced here, it goes into the gallbladder to get concentrated. So now the gallbladder contracts, pushes the bile into the cystic duct, it comes then into the bile duct, and CCK also causes this sphincter of Oddi to relax, so that both bile and pancreatic juice can come into the duodenum. Okay? Let's look at the function of these two juices. Bile, remember, is not an enzyme. It does not actually digest anything. What it does is it emulsifies fats. Emulsifying means it breaks it down into smaller particles. And by breaking it down into smaller particles, it increases the surface area for action by lipase. Lipase, if you remember, lipase, lipid, is a fat digesting enzyme. So if you had one big blob of fat like this, with no bile salts, this lipase enzyme, and let's say I just draw it like this, what it will do is it will be able to act on the surface here. Whereas when you add bile salts to it, this big blob of fat is actually broken down into smaller particles like these, and you have bile salts attached to them. So now lipase has a larger surface area on which to act, and so it can act on all these particles and it can digest fat really well. So that's the purpose of bile, to emulsify or break it down into smaller particles so that it can be acted upon by lipase. It also facilitates cholesterol and fat absorption. You will see once fat is broken down into its constituent parts, which is fatty acids and glycerol, it has to be taken into the intestinal cell and then go into the lacteal. By itself, fat cannot go. It actually is bound to bile salts, and that's what it forms something known as micelles, which we'll see, and that's what helps it to get into the intestinal cell. Bile also keeps cholesterol, bile salts keep cholesterol dissolved in bile, and whenever the ratio between bile salts and cholesterol changes, then you can get deposition of, these salts or cholesterol, and that's when you get gallstones. So when people get gallstones, it's because this ratio has been disturbed. And secondly, and lastly, in bile, you have bile pigments, and one of the pigments called bilirubin is the one which gives feces and urine its color. So, the yellow coloration of feces and urine is due to what is called urobilinogen and stercobilinogen. So that's the pigment which is present. Let's look at pancreatic juice. Number of enzymes. So collectively, the protein digesting enzymes are known as proteases. The carbohydrate digesting enzymes, very similar to salivary amylase. Here we call it pancreatic amylase. Then we have lipases, which digest fats, and they need the bile salts to be present. And if bile salts are not present, fat is not digested properly, neither is it absorbed. So what happens is it gets passed out in the feces. So the feces are very, very greasy. And then pancreatic juice also has a lot of bicarbonate ions, and the function is to neutralize the acidic kind which comes in. And I said that cholecystokinin and secretin, which is released by the intestinal cells, that helps to control this bile secretion, bile and pancreatic juice secretion. So let's take a look here.
Welcome to the tutorial. This time we're going to look at how CCK, which is a hormone, causes bile release from the gallbladder. So first of all, a little bit of digestive anatomy. We've got the stomach here, which has the esophagus that leads into it, and coming out is the first section of the small intestine, the duodenum. Off to the side is the liver here, and underneath the gallbladder, and then the bile duct, which leads from the gallbladder into the duodenum. And that's what carries the bile. So over to the stomach again, when you have fats in your diet, as we inevitably do, a lot of the lipids are clustered together in sort of globules, illustrated by this. Whereas single lipids that make up the globule would be represented by just one L here. Now the hormone CCK is going to be involved, and you'll see how in a second. It stimulates the release of bile, and bile is going to go in and cut this lipid cluster down, so we have single lipids, and then lipase, the enzyme, will act in the duodenum and break down the lipids for us. So let's watch the animation. So the lipids are in the stomach, they come down and rub the wall of the duodenum. The hormone CCK travels through the blood to the gallbladder. The bile comes out and splits the large lipids into small ones. And now these small lipids can be acted on effectively by lipase. So the action of bile there doing the splitting part is called to emulsifying the lipids. Okay, so I hope this helps you.
So again, you can see all it did was break it down into, it's like almost, if I could, though not correct, mechanical digestion. I mean, there was no kind of churning or anything, but they kind of just mechanically break it down into smaller particles. So there's no chemical digestion that they do, that bile. That's why bile is not an enzyme. Then, just as in the stomach, when food was being digested, remember churning movements were occurring so that you can completely sort of mechanically break down the food into smaller particles and, help with the digestive process. Similarly, in the stomach, in the small intestine is kind of broken down into tiny sort of segments, and each segment kind of swishes the contents back and forth. So look at the beginning. So here is one segment. So you have a little bit of food chyme here and another part here. So now it is swishing it back and forth. Notice how it's going back and forth so that this is thoroughly mixed, and then now you get kind of a complete mixture here. So this part is known as segmentation, which is mechanical digestion. After this, this segmentation slowly kind of allows it allows the mixing of food, but it also allows slow movement of the food towards the ileocecal valve, because it's now going to have to move towards the large intestine. True peristalsis, where the wave passes from above downwards, like you can see here, just pushes the food forwards like this. True peristalsis actually begins only after the nutrients have been absorbed, okay? So here let's look at both segmentation and peristalsis, and you'll be able to see this quite well in this video. So notice the segmentation occurring here. So see how it's kind of broken into tiny segments as going back and forth between. So you can see the mixing that's occurring here. So that was more on segmentation really, but peristalsis would just cause a forward movement. Now let's look at absorption. So we've broken the food down into the smallest particles. So by the time chyme actually reaches the ileum, as I mentioned, most of the absorption has taken place. So here we have water, electrolytes and nutrients which are absorbed, and active and passive transport mechanisms are used. So for carbohydrate and proteins, some of them are absorbed actively. Fat and water is mainly by passive transport. Simplest forms are used. For example, in the case of carbohydrates, it will be monosaccharides. So in the case of carbohydrates, they'll be absorbed in the form of monosaccharides such as glucose. In the case of proteins, they will be absorbed as amino acids. In the case of fats, fats are broken down into fatty acids and glycerol. But these cannot be absorbed as such, so I'll show you what happens in the case of fatty acids and glycerol. But glucose and amino acids enter blood vessels. Fats enter the lacteals, which are in the center of the villus. So here, if you look at this picture here, this is a big fat globule. So you can see how bile salts, when they act, they break it down into smaller particles. This is the emulsification, so that helps the lipases to act on it. So lipases break it down, and they break it down into fatty acids and glycerol. This then again combines with these combined with bile salts to form structures which are known as micelles. And in order to enter the epithelial cell, it has to be attached to the bile salts. That's why bile salts, we say, help in the absorption of cholesterol and fat. So once they get in, then the fatty acids and glycerol break away from the micelles and they join up together again to form fats, and then they are carried in the lacteals. And remember, the lymphatic system in the end goes into the venous circulation. Okay? So here in a villus, let's look at the absorption.
The lining of the small intestine is characterized by numerous circular folds called plicae circularis. The plicae are with finger across. The villus contains a network of capillaries which surround a lymphatic vessel known as a lacteal. The epithelium of an intestinal villus consists of cells which are with microscope. This succession of folds and projections increases the intestinal lining. Carbohydrate digestion enzymes in the small carbohydrates are absorbed by bile and then enter the capillary. Fat digestion occurs primarily in the small intestine. Fats are digested into the epithelial cells of the villus. The fats are formed into clusters called chylomicrons into the lacteal. Lymph carries chylomicrons away from the villus. Protein is complete in the small intestine. Proteins are broken down into peptides, then into amino acids. These are absorbed into the villi, then into the capillary.
So here's a really good review of digestion and absorption. So kind of make sure you get it. So if you look at carbohydrate digestion, and it tells you which are the enzymes which digest it, where are they produced, and how is it absorbed. So if you look at it first, carbohydrate digestion begins in the mouth, where this big complex carbohydrates are broken down into smaller carbohydrates. Then in the small intestine, it is completed by pancreatic amylase and the brush border enzymes. And so these enzymes, like I said, have names like lactase, maltase, sucrase, digesting maltose, lactose, or sucrose. And so they are broken down into their simplest forms, which is glucose, galactose, or fructose. This is then absorbed into the blood circulation through facilitated diffusion. Next is protein digestion, begins in the stomach, where pepsin is secreted in an inactive form and needs HCl. And then again, you have pancreatic proteases, whose names are things like chymotrypsin, trypsin, and we have brush border enzymes as well, which have names which end with like a peptidase, aminopeptidase, dipeptidase. So they break down the polypeptides into smaller molecules. So finally you get amino acids. They again go into the bloodstream. Then when you look at fat digestion, lingual lipase and gastric lipase, this lingual lipase can act a little bit in the gastric in the stomach, but it kind of gets broken because, remember in the mouth the pH was almost neutral, in the stomach it's acidic. So these really don't have much to do. The main digestive enzyme which acts on fats is the pancreatic lipase, which depends on bile to help digest it. And then this goes into the lacteals. And then, as I said, these were the three main food groups, but then you have nucleic acids as well. And so you have nucleases, which the pancreatic juice contains, which break it down. And, those are also absorbed into the bloodstream. So really good slide, which kind of gives you everything in a nutshell. So let's answer a few of these questions. Which of the following statements is not true? Yes, this is wrong. Bile does not digest fats. Remember, it only helps in the digestion. It cannot, only an enzyme is capable of digesting. It only helps, it doesn't digest. It does break down fat into smaller particles. So this is true. This part is true. Okay, here. Apart from causing intestinal, pancreatic and bile secretion, what is the other function of cholecystokinin? Yes, it causes relaxation of the gallbladder, relaxation of the sphincter of Oddi and contraction of the gallbladder. It can't make the pH neutral. Remember, it's a hormone. So it can't make the pH neutral. To make the pH neutral, you need something like bicarbonate or you need mucus. Okay? So this is the correct answer. Okay, what are brush border enzymes which act on proteins and carbohydrates? What are they produced by? Very good. By intestinal epithelium, not the Brunner's glands. Remember, the Brunner's glands were glands present in the submucosa. Brush border enzymes are part of the epithelium and they form that fluffy, fuzzy brush-like appearance. Okay, which of the following statements is false? Okay, the correct answer is the fourth one. Now let's look at this. The epithelium does produce enzymes and other substances responsible for chemical digestion. Remember, that's where it was, that's the epithelium cell. We just saw the brush border enzymes are part of the epithelium, right? In the stomach, for example, the parietal and chief cells were part of the epithelium. So that's where the secretion occurs. The muscularis externa is responsible for muscular movements. The glands in the submucosa, they do help in digestion. It doesn't say they cause digestion, it's a help in digestion. Remember the Brunner's glands, they help because they neutralize the acidity in the duodenum so that now you have an alkaline medium in which pancreatic enzymes can act. So they do help in digestion. Absorption of nutrients occurs through the epithelium. So if you take a cross section of the digestive tract, and in the case of the small intestine, if this is the lumen, so imagine I'm just going to draw the lumen like this. So this is the lumen, here's that muscularis mucosa. So this area here is submucosa. This is muscularis externa on the outside. And this is the serosa, the outermost layer. So your food is being digested here. And if I just take a little section through like here, so you can see here's the villus, the crypt, and here's submucosa, here's muscularis externa, and here's the serosa. So this is the lumen. So this is where food is being digested, has been broken down. These are the epithelial cells, and you just saw that video too. So you saw how from here they pass through the epithelial cell and then go into the blood vessel. So if you think that absorption of nutrients is not going to, if you think that this answer is true, then what you're saying is that from here somehow, from the lumen, somehow it manages to go outside, and then from here it's going to be absorbed. You see that? So that's a false statement, okay? So that's why this is the correct answer to this. All of the other things do happen. So absorption is through the lumen, not through the serosa. The glands in the submucosa, so this is the false statement. The glands do help, and the epithelium is the one which produces enzyme and other substances. By other substances, I mean like mucus or HCl, which help in the digestive process. Let's look at the large intestine. As we said earlier in anatomy, it's called large because the lumen is large, not because its length is larger. Its main function is the absorption of water from undigested food. So it's the absorption of water from undigested food, because whatever has not been absorbed in the small intestine now passes into the large intestine. And all that it contains is some water that it can absorb. So that's why it takes long to pass through the large intestine. The large intestine had those haustra, you remember the circulation, so that the undigested food kind of stays in each circulation for a while so that it can absorb water. There are some electrolytes which may be absorbed here, and there is elimination of feces. In the large intestine, you have bacteria, which we mentioned. This was called the bacterial flora. These are very important. They are symbiotic with us, so they take in the undigested food. You know, sometimes when you eat, we have a fiber, for example, cannot be digested by us. We don't have the enzyme cellulase to digest it, but some animals do. So when you eat fiber, it just increases the bulk of your contents, and that helps in the contractions of the large intestine. But that undigested fiber, which is carbohydrate, is acted upon by the bacteria. So the bacteria use it for their nutrition. And in turn, what they do is they do a lot of things. They produce vitamin K and B complex. In fact, the amount of vitamin K that is produced is enough for our clotting factors. They also help in the immune response. They actually help in our immune response. They do produce some short-chain fatty acids which may be absorbed in the large intestine. And sometimes when you give people antibiotics for a prolonged period, and especially what are known as broad-spectrum antibiotics, when you give them antibiotics which can act on a variety of bacteria, not only does it kill, you're giving a person antibiotic because there's some infection in the body. So it kills the infection-causing bacteria, but what it does is it also kills off our bacterial flora. So quite often when this is done, people end up with, you might see people on long-term antibiotics, they might end up with mouth sores, because B complex is needed for cells which divide very fast, so your GI tract divides very fast. These people may have diarrhea. So, sometimes people on antibiotics end up with diarrhea. You may have this normal good bacterial flora that keeps other bacteria at bay. So it prevents the over-colonization by the other harmful bacteria. But when they are killed off by the antibiotics, you'll find that people end up getting, with overgrowth of these other harmful bacteria. And one such bacteria which tends to populate quite frequently is something known as Clostridium difficile. So that C. diff, as it's called, when people are on broad-spectrum antibiotics, especially in hospitals, stay for a very long time, they get severe diarrhea because the good bacteria are gone, and this C. diff, which is normally kept quiet, now kind of tends to grow, and that can cause diarrhea. And nowadays we talk a lot about probiotics. Probiotics are nothing but live cultures of the very same bacteria which colonize our gut. And, when people have don't have adequate quantities, then, you have to take these probiotics either as in yogurt or, you take them as pills. And I'll just show you a video on that. The other thing that happens in the large intestine is the defecation reflex, which is mediated by the parasympathetic system. You get about 2 or 3 waves which pass all through the large intestine, and there is an urge to defecate. If the final defecation is under voluntary control, because remember there were 2 anal sphincters, an involuntary and voluntary sphincter, so the time and place is not right, then you won't defecate, and then the wave passes off and the urge to defecate passes off, and then you have to wait till the next wave comes. So let's look a little bit at probiotics, since there's so much talk about it nowadays, and it's really something that most of us need.
In most cases, the organisms in our food get mashed around in the mouth, then hit the roller coaster that rapidly into our stomach. The stomach is a particularly inviting place. Acid is produced in large quantities when we eat. This helps to protect our body and to break down, and it kills the bacteria that we ingest, including many of the milk fats present in dairy products. However, some bacteria, such as these probiotic lactobacilli and bifidobacteria, can survive the acid by having a protective shield, shown as protection provided by or by leaving the stomach rapidly with the liquid part of the meal. More than half of the world's population, and especially bacterium pylori, in their stomach. Some strains of this bacterium are genic, acquired from several sources, contaminated water. This bacterium can burrow into the walls of the tissue and produce a toxin that damages cells, causing painful ulcers and sometimes cancer. Treatment can helicobacterialization, but in others, the bacteria turn again. Probiotic may have a reduction in these helicobacterias, perhaps by attacking them with antibiotic-like weaponry called bacteriocins, or dislodging them. However, more researches and trials will determine from ingesting certain types of probiotic bacteria. The dead organisms, along with the probacteria and surviving fermenter microbes, move towards the intestine, where they come into contact with bile salts, which kill the milk. Many probiotic bacteria can resist these harmful effects and survive and continue their journey. Next, takes the back down through tissues into the small intestine. This region of the body is usually over 30 feet or 9 meters in length. Structures called microvilli, which absorb nutrients, form a defense disease. The small intestine is home to billions of bacteria, microbiota, including bifidobacteria and lactobacilli, which play a critical role in keeping us healthy through degrading food, counting bad bacteria, and modulating the immune response. Questions need to be studied, such as bacteria leak each other and influence the way we feel, as whether probiotics can prevent or alleviate certain diseases.
So it went as the small intestine, but then they reach the large intestine, where that's where they kind of use the fiber and the undigested food, and that's how we have a symbiotic relationship with them, and that's where they produce the B complex and K, which is absorbed into our bloodstream. So now let's look a little bit at metabolism. So you ate food, you digested it, and then you pushed it out. So now let's see what are What is the fate of all the nutrients that you absorb? So there are processes in the body which are breakdown processes and processes which are building processes. So if you look at it, anabolic, the word anabolic means to make something or synthesis. Or you'll often see the word genesis associated with it. And catabolics is breaking down. So usually breakdown, you have the word lysis associated with it. In our body, anabolic and catabolic processes occur side by side. Because, so for example, when you have proteins broken down into amino acids, that's a breakdown, right? So that's a catabolic process. Those amino acids go, they're absorbed into your bloodstream, and then when they go towards the cells, they join up together to form proteins which are needed to make, for example, maybe hemoglobin, maybe gamma globulins, and things, fibrinogen, all of these things. So you see that you broke it down only to kind of remake something else. So anabolic and catabolic processes occur side by side in our body. And so let's first start with carbohydrates and what is the fate of what you eat and what is absorbed, what happens to it. First of all, carbohydrate forms those monosaccharides which is absorbed into your bloodstream. The main purpose and the first source that your body wants to use for energy is glucose, which is a monosaccharide. And the idea of using glucose is to generate ATP. So you all know if you suddenly want quick energy, you drink something sweet so that you can take that glucose and it can be broken down. Then if there is extra glucose left, because you don't only eat that much carbohydrate which provides you ATP for that day's activity, you can't measure it that accurately. So there's always some carbohydrate left. That carbohydrate is converted into glycogen. So this is glycogenesis. So you can see it's an anabolic mechanism. It is converted into glycogen and it goes and gets stored in the liver and skeletal muscle. When your body needs it, let's say there isn't enough carbohydrate coming to it, then the body can call on these reserves and break it down. So that glycogen is broken down. So this is catabolic. So see the word glycogenolysis. So glycogen is broken down. So remember lysis was for breakdown. So glycogenolysis when needed. This process of knowing that if you eat a lot of carbohydrate, it can go and get stored as glycogen is used by athletes, and this is perfectly legal. They do something which is known as carbohydrate loading. So what they do, especially before an event which requires where you require a lot of energy, let's say before a swimming event or before a tennis game, let's say they have a match 2 days from now. What they'll do is they will eat their diet on those 2 days will be high in carbohydrates. So eat a lot of bread and eat a lot of pasta. So what happens, that goes into your body, it's broken down into glucose. Some of the glucose is absorbed. What is left is then stored in the liver and skeletal muscle. So it's not weeks before, it's just a few days before. So that goes and gets stored in the liver and skeletal muscle. And then when they are playing at that time, see that glycogen, because their body needs a lot of energy, that glycogen can be broken down, made into glucose, and they get energy. So this is perfectly legal. This is not like erythropoietin or something like that. Okay, so this is known as carbohydrate loading. And if you ever ask any athlete what their diet is, especially swimmers, people who are going for a sprint or tennis or things like that, what they eat, they'll always tell you a diet rich in carbohydrate. Now let's say your body had more carbohydrates, so you made all the glycogen you could and you stored it in the liver and skeletal muscle. What about the extra carbohydrate? That can be used for protein and lipid synthesis. So that's why you see if your diet is too high in carbohydrate, you can end up forming fat, and that's how you end up putting on weight. Okay, so that's why they say a diet high in carbohydrate results in fat deposition, because the extra carbohydrate is made into lipids. Let's see proteins. So proteins are absorbed in the form of amino acids. So you first form amino acids, and then these are used for making protein-containing substances in the body. I gave you some examples. So you have, for example, hemoglobin, which is the pigment in blood. You have myoglobin, which is a similar pigment in muscle. You have gamma globulins, which are antibodies. Then you have clotting factors like fibrinogen, prothrombin, all of these, right? Albumin, which is responsible for maintaining the osmotic pressure. So you can see all of these are the amino acids I'm going to manufacture these protein-containing substances. Your your muscle cells, they also contain protein. Your skin, keratin, is protein. So you can see how so many substances in the body contain protein. So this is what it's used for. It's also used for growth and repair of tissues and, like we said, synthesis of substances containing protein. Then, if carbohydrate is not available, proteins can be used for ATP synthesis. And proteins can also be used to make glucose. That process is known as gluconeogenesis. So as you can see the word, and we went over this in endocrine system, gluco means glucose, neo means new, and genesis is to make. So to make glucose from new sources or non-carbohydrate sources. So extra proteins are, extra amino acids are used to make glucose. So that's called gluconeogenesis, or they may also be utilized to make fatty acids and glycerol. So they can be used for lipogenesis. Next is lipids. When they are broken down, you have the formation of fatty acids and monoglycerides. They join up together after they get into the lacteals, they form what are called triglycerides, which go bound to proteins, and these are called lipoproteins. So in your notes, I've given you examples of, low density lipoproteins, LDL, which is often called the bad cholesterol. Then you have VLDL, very low density lipoproteins, and you have something called high density lipoproteins. High density lipoproteins are the good fat, as we say. And then we also have cholesterol. So all these are carried bound to proteins. So they're carried as lipoproteins and taken to different areas of the body. Fat is stored in adipose tissue. Now, fat is good because it's needed in your body for making of cell membranes. Remember, cell membranes are lipoprotein in nature. Myelin sheath is something which is fatty in nature. Many hormones are steroidal, again need fat, so it's important. Fat is an extremely good source for ATP production. If glucose is not readily available, then the body calls on fat. So during starvation, for example, people tend to lose a lot of weight because first the body's sources of glucose and glycogen are used. And then protein breakdown occurs, so they lose weight through muscle. And then you can see that, the skin and everything starts sagging because the fat from all over the body is broken down in order to produce ATP. So even you'll see that if someone is starved for an extremely long time, even the fat inside and around organs, not inside the organ, but around organs, around blood vessels, that fat goes. And when we do the urinary system, I'll tell you there's a condition where the kidney drops. The kidney is kind of surrounded by a layer of fat. And when that fat is depleted, the kidney actually drops from its original position down below, and that can cause kinking. And that's seen, and it used to be seen in concentration camp, people who were interred in concentration camps because they didn't get food to eat, right? So the body had somehow to make energy. So it used up glucose reserves, glycogen, proteins, and then fats. So, fat is a very good source to make ATP. And then fat can also, like proteins, produce glucose when needed. So from fatty acids, you can get glucose production. So gluconeogenesis occurs. So let's take a quick look at this fate of nutrients.
Latte or Olay, cozy or cool, or exactly the way you want it, until boom, it's better.
So now you've absorbed all this food. You've eaten this hamburger or whatever it is you eat, maybe tofu. All the broken down small intestine absorbed into your blood and into your lymph system. The question now is, where does it all go? What's next? And we actually have and the other is called the post. I like to call them when you're not hungry and when you start to get hungry. You're obviously past absorption. And I want to start out with the absorptive state. Now, let's take a look at this chart here. You know, in the absorptive, you've got all these nutrients floating around in your blood. Okay? And you've got some of these nutrients floating around in your and eventually going to end up in your blood. Let's take a go. And let's start out with acid. So, that's proteins. That's just the way it goes. You see, we can't store amino acids. Amino acids are made to, and this amino either got to do something special with them, which is later, or your body. Amino acids is the simplest one. They come on down, they go to all your tissues, through your blood, they go to the liver, happens synthesis. You get DNA, RNA makes protein, the amino acids are put together, you get protein synthesis, you get protein synthesis. Amino acids, simple. It gets a little bit more when we get saccharides. And the monosaccharide we're going to that for energy in respiration. And we'll look back at those respiration lectures to really review that because it's a pretty complex process. So what's going to happen with glucose? Again, one thing in particular, but two things could happen. The first thing is it's going to get shunted to the liver. Well, stops after the intestine is blood is going to get scooted to your liver. Way over a dozen functions, many of which are going to occur in the absorptive and the post-absorptive state. So what's going to happen there? Well, when it goes to the liver, depending on the levels of glucose, to end up stored as glycogen. Now, one thing I do want to mention, remember insulin, okay? Well, and that's going to do these things. In fact, it's going to glucose used during its absorptive state, and in this case, it's going to get stored as glycogen. Now, does that mean all of your glucose stays in your liver as starch, this long complex starch called glycogen? Well, of course not. You take in a lot of glucose over the course of the day or in the course of a meal. So a lot of dependent to all your other tissues. Hey, and once all those other tissues, what's it do? Look back at those respiration lectures. Peak.
Okay, so he didn't finish lecture here. So it's chart, you know, where it all went. So let's look at this. Which of the following is a catabolic reaction? Yes, remember, whenever you see the word genesis means to make, anabolic is to make. When you see the word lysis, means to break, catabolic means to break. So glycogenolysis is a catabolic reaction. So this is a small slide that I put in today, which just shows you what happens in metabolism. So all of those nutrients which we saw, and you know, the final thing was to provide energy for your body. So when you look at all these organic molecules that you've taken in, amino acids, lipids, and simple sugars, they get into the cell. So they form a nutrient pool in the cell. Some of it is used up for catabolism, some of it is used up for anabolism. And so initially there is processing to produce ATP occurs in the cytoplasm where not much is produced. Then next it goes into the mitochondria where the final processing occurs. And in the mitochondria, when say glucose is broken down, so I'm just using glucose, you get 60% is taken out as heat, which is what keeps our metabolism. Remember, your basal metabolic rate. So 60% liberated as energy, 60% of it is actually as heat energy, 40% is ATP. And this 40% which the body produces is used for anabolic processes. Anything anabolic requires energy. When you want to build something, you need energy. It's easier to break it down. You don't need energy, right? Imagine if I told you to break down a house, you don't need that much energy. If I told you to build a house, you'll need a lot more energy. So this ATP which is produced is the one which synthesizes new molecules. It's also needed for things like active transport, which you did in ANP1, your endocytosis, exocytosis, phagocytosis, mitosis, all of that, okay? Now here you'll see that all of these fatty acids, glucose, glycogen, which is broken down as to glucose, and amino acids, they go through a series of processes. They go through glycolysis first, then they go into Krebs cycle, and from there they go into something known as the electron transport chain. And it's this electron transport chain which is the final place where maximum ATP is produced, okay? So this is more biochemical. So, I could kind of go on ad nauseum. It won't enter your head too much. So what I want you to know is, one is this part here, and the second is that the glycolysis, the Krebs cycle, and then the electron transport chain, which I've mentioned in your notes. From that, you can see that it's in the electron transport chain that maximum ATP is manufactured, and that's what our cells actually use to manufacture ATP. And this is what it shows you. So here you can see in the stages of metabolism. So proteins, carbohydrates, and fats. Notice how proteins are broken down into amino acids, carbohydrates into glucose and other sugars, and fats into glycerol and fatty acids. This pink or color is anabolic, and the blue is catabolic. So when they are broken down, this is a catabolic process. Amino acids end up forming proteins in the body, so this is an anabolic process. Glucose is stored as glycogen. This is an anabolic process. Glycerol and fatty acids form fats, which is an anabolic process. All of these, if you notice, they go through glycolysis. Then they form an intermediate compound, which is called acetyl-CoA. Then they get into the next phase, which is Krebs cycle. So at each point, a little bit of ATP is released. And then from the Krebs cycle, they go into the mitochondria, where you can see they produce hydrogen ions, and getting into the mitochondria through the electron transport chain, that's where maximum ATP is produced, okay? So what is this electron transport chain? So again, if you look here at the mitochondria, so you can see glycolysis occurs outside the mitochondria, you get very little ATP produced. This, through glycolysis, the next phase is it goes into the Krebs cycle. You get a little bit of ATP. The glycolysis produces something known as pyruvic acid, which goes into the Krebs cycle, and that produces a little bit of ATP. But from here, it actually goes into the mitochondria, where you have proteins which are present inside this folded membrane. Those proteins catch electrons and they transfer it, and finally they end up transferring it to oxygen, and they produce a lot of ATP, as you can see here. A lot of ATP is produced through this electron transport chain, okay? So let me show you this. Let's go back. Electron transport chain. So this will kind of explain to you what exactly is happening. You don't need to worry about the enzyme names.
You know the feeling.
Most of the energy harvested from organic molecules during glycolysis and the Krebs cycle is stored in NADH. DH2 molecules give up their high-energy electrons in the third phase of respiration, electron transport in ATP synthase, where most of the cell's fuel is produced. Transport is in our molecules, mostly proteins, built into the end of the mitochondria. NADH energy electrons to the first complex in the electron transport. The electrons giving up their energy as they are pulled from NADH for oxygen. The energy given up by the flow of electrons is used to pump hydrogen ions from the mitochondrial matrix to the intermembrane space. Oxygen captures the ions in the very last step in electron transport. The last comes to an oxygen atom to hydrogen ions, forming water. Electron transport has used the energy of moving electrons to pump hydrogen ions into the intermembrane space. This buildup of hydrogen ions, like water behind a dam, the potential that was originally in the bonds of glucose molecules. The backed up hydrogen ions give up their energy when they diffuse a special protein compound in the membrane called synthase. As hydrogen ions flow down their concentration, ATP captures their energy to make ATP.
Saw how there were so many hydrogen ions up, so you can understand how when come down, they capture the end, and that's how you a lot of ATP produced. So what I really just want you to know is in glycolysis you kind of get 2 ATP, in Krebs cycle you get 2, in this you get 32 really, and it says 34, but 2 are used up for other things. Okay, so the maximum amount of energy is generated in this electron transport chain. So let's answer this last question. So let's say someone's not eaten for 6 days. Which of the following reactions do you think would not be of importance in providing ATP? So think what the body is going to do at this time. And which of these is the body not going to do? Okay, the correct answer is number 4. And let's see, everyone here agrees that glycogenolysis will occur. Obviously, you from glycogen break it down, you will produce glucose, that will produce ATP. You will have gluconeogenesis, because remember, what is gluconeogenesis? Production of glucose from non-carbohydrate sources. So if glycogen stores, she's not eaten for 6 days, so in a few days her glycogen stores would be depleted. So now she needs energy. So the proteins and fats would be used to making glucose, and that will produce the ATP. Fat will be broken down so that ATP can be produced. At this time, you don't want protein synthesis to occur, because this is going to use up so much ATP. And what's the point of making proteins at this point? You're actually going to break down proteins. You're not going to be making proteins, right? So protein synthesis is something that's not going to occur at this time. You know that when someone is starved, muscles become smaller because protein is broken down. Muscles are made up of protein. Fat is broken down. They get thinner. They lose fat, right? So all these 3 will occur. This is what is not going to occur. Okay? Okay.